Selamat siang semuanya. Suara saya terdengar dengan baik?
Terdengar.
Terdengar ya. Ya. Oke. Bagaimana kabarnya? Luar biasa. I hope you all in a good condition ya. Sepertinya sudah hadir bersama kita Prof. Lee Seung Bao. Ni hao lao sư.
Hello.
Hello lao sư.
Good afternoon.
May I call you lao sư or Prof. Lee?
It's fine.
Oke. Ya, jadi Prof. Lee sudah bergabung bersama kita ya.
It's fine. Thank you.
Ya, kita akan segera mulai seminar kita pada siang hari ini. So, as we are about to begin, I would like to see your cooperation in completing this seminar. Thank you for your kind cooperative and attention. Oke, jadi nanti kalau selama Prof. Lee memberikan seminar ya, terus kemudian nanti kita ada sesi tanya jawab, nanti silakan menggunakan fitur raise hand, kemudian bisa melalui chatnya seperti itu ya, jadi bisa langsung berinteraksi dengan launcher. Oke. Good afternoon everyone. Yang terhormat Direktur Akademi Farmasi Putra Indonesia Malang, Respectable the Director of Pharmacy Academic of Putra Indonesia Malang, Honorable Mr. Prof. Lee Seung Bao, PhD, as speaker, and respectable all participants of this seminar. Pertama-tama, marilah kita panjatkan puji dan syukur kepada Tuhan, karena atas berkat dan rahmat-Nya, kita dapat berkumpul di sini bersama-sama tanpa hambatan dengan sehat. First of all, let's thank the Almighty God who has given us some mercies and blessings, so that we can attend this agenda without any trouble and obstacles in the seminar of improving lecturer and student competence through guest lecturer. I would like to introduce myself. I, Andini, will be your master ceremony today. Selamat datang di seminar hari ini dalam rangka Improving Lecturer and Student Competence through Guest Lecturer. Welcome to today's seminars. We would like to thank everyone for making time to attend this remarkable seminar. Ladies and gentlemen, on this nice occasion, let me deliver the structure of the event today as follows. First, opening. Second is singing Indonesian anthem. Third, welcoming speech. 4, presentation. And 5 and 5, closing. Ladies and gentlemen, before we come to the main session, let's open this event by praying first. So the event that we will hold on this day will run well without any obstacle at all. Pray based on individual belief. Pray begins. Pray done. Now we're gonna go to the next agenda. Singing Indonesian anthem. Next agenda will be a welcoming speech. Allow us to invite the Director of Pharmacy Academy of Putra Indonesia Malang that will be represented by Ms. Ambar Vidyasari MP to deliver her welcome remark. Please welcome.
Thank you, Miss Andini. Okay, how are you today, Prof. Lee? Nice to meet you, Prof. Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Best wishes to all. The Honorable Prof. Lee Sumbo, the Honorable Director Academy Pharmacy of Putra Indonesia Malang, a distinguished lecturer of Academy Pharmacy and Academy Food and Pharmacy Analyst Putra Indonesia Malang, and our beloved student for Taipei Medical University of Academy Pharmacy and Academy Food and Pharmacy Analyst Putra Indonesia Malang. Alhamdulillah, we dedicate our deepest gratitude to Allah SWT because for His grace and guidance we are able to attend this valuable grace lecture in good health. First of all, on behalf of the institutional leader, I would like to thank you very much, Mr. Prof. Lee Sumbo, who has spared his time and share his knowledge with all of us here. Welcome to our Putra Indonesia Malang campus. Nice to meet you. Hopefully the sharing of knowledge today is beneficial and can provide inspiration for our lecturer and student. Hopefully also this activity doesn't end here. We hope for the great opportunities to have future networking for the lecturer of Aka Pharma and Akwar Putra Indonesia Malang with the lecturer of Taipei Medical University. I will be very beneficial for the development of not only in education, but also in research for our lecturer and students. Once again, thank you very much, Prof. Lee Sumbo. We apologize if there are some unpleasant things to the lecturer and students. Congratulations on following and listening to the extraordinary knowledge. Stay excited and happy. Thank you very much. Wabillahi taufik wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you
Okay, I
very
give
much.
it to Miss Andini.
Yes, thank you very much, Miss Ambar, for your delightful speech. Okay, ya, as Mrs. Ambar said before, ya, stay healthy and stay focused on this guest lecture. Okay, ladies and gentlemen, now we proceed to the seminar agenda. This presentation will be delivered by Mr. Li Shengpao, PhD. Ni hao, Lao She. And also, this agenda will be guided by Mr. Kiantoro Andi Setiawan, Magister of Linguistics, as the moderator. We invite all the distinguished speaker and the moderator to take over. Please, waktu dan tempat dipersilakan kepada Pak Kian.
Okay, thank you, Ms. Andini, for the nice speech. Okay, good afternoon, everyone. Thank you for joining this guest lecture today. Today we have Mr. Sumbauli from Taipei Medical University, and my name is Kian. I will be the moderator of this guest lecture today. Now, before Mr. Sumbauli deliver his presentation, let me introduce you. Let me read the curriculum vitae of Mr. Sumbauli. Mr. Sumbauli obtained his Magister of Science from the Department of Life Science, National Tsinghua University in Taiwan in 2002. And he also obtained his PhD degree from Graduate Institute of Life Sciences, National Defense Medical Center, Taipei, Taiwan in 2009. He joined postdoctoral fellow Genomics Research Center in Academia Sinica, Taipei, Taiwan in 2009 until 2011. And he also joined postdoctoral fellow BRIC, University of Copenhagen, Denmark in 2011 until 2016. In 2016 until 2020, he became the assistant professor, master program in clinical pharmacogenomics and pharmacoproteomics in Taipei Medical University. And in 2020 until 2021, he became the assistant professor, PhD program in drug discovery and development industry, Taipei Medical University. And for now, in 2021 until present, he became the associate professor, PhD program in drug discovery and development industry, Taipei Medical University. His research interests mainly on chromatin replication, genome stability, cancer biology, and drug discovery. Without further ado, let me invite Mr. Sumbauli to deliver his presentations. Time is yours. Please.
Thank you very much. Okay, let me share my slide first. Okay, can you see my slide?
Yes, clear.
Okay, great. Thanks for the invitation to me to come to this lecture. I'm really happy to be a speaker, and I would like to deliver some knowledge and some research idea to you. And also, as mentioned, today's talk will focus on. I will focus on the high throughput screening, but about this experiment, I will put it in the end of this talk. Okay. So first of all, I would like to introduce myself again. Yeah, as mentioned by the moderator, that I'm now in a PhD program of drug discovery and development industry at TMU. Before I joined this program, I was in the master program of clinical genomics and proteomics. Yeah, and if you look at my publication, you can see that before I joined the TMU, I was working on the basic research on the mechanism of the DNA replication in human or in virus. I'm worked on this field for a really long time. But when I joined the TMU, I would like to use my knowledge to think about whether I can translate this knowledge to the drug discovery. Yeah, so because as I mentioned that I was studying the mechanism of the DNA replication for a long time, and I also learned a lot of things, knowledge in mitosis or other phases of the cell cycle. So I'm thinking that when in the TMU, I would like to work on the cell cycle control. I would like to focus on the cell cycle control and think about where we can target this kind of control to discover better small molecule that can against the cancers, or we can develop better strategy, better combinational strategy to treat the cancers. Yeah. So within this year, we have published several medicinal chemistry paper and also some pharmacology papers in these fields. Yeah, and just for your reference, and today I will talk one of these papers, and also I will also introduce our current experiment, current studies that have that is now is currently submitting to the Genome. So before I talk about our study, I would like to tell you some concept. Probably you never heard about it. Yeah, and this slide, I would like to show you that this is the table showing the top pharmaceutical company, how many monies they have budgets they have spent in the research and the development department. Yeah, and you can see. Sorry, let me change the. Change to this. Yeah, so you can see that in the 2021, the top company is Roche, and then Pfizer, and also Novartis. Yeah, so you can see that they have spent several billion on R&D department, right? And you can also see that in 2020, the year 1 year before, they spend much more budget on this department, right? Now, I think that's because of the COVID-19 issues, so they reduce the budget on this R&D budget. But still, you can see there's still a lot of money on this department. So they still, although they are already a big company, they still spend a lot of money on the drug development, right? So, so this field would be still a good field for all the all the students. They can move on, yeah. So to develop the drug, we have to think about what target we would like to focus on, right? We can use the genomics, proteomics, or other kind of the strategy to search and to think about which target is better for the drug discovery, right? When we have a target, then we need to think about how we can analyze this target activity, right? We need to develop an assay to analyze this target activity. And then we even can develop a high-throughput screening assay to evaluate which compound, which drug can modulate this target. Okay, yeah. So after we use this assay to find a compound that can modulate, probably inhibit this target activity or activate this target activity, right? After that, we got one lead compound, right? We have a lead compound which can modulate our target activity. And then we can, through the medicinal chemistry method or probably some in silico screening, to optimize this compound, to modify this compound, to let it become more potent or less toxic to the normal cells. Okay, and then when we have several candidate compounds, then we need to go for the in vitro study in the cells or in probably in other diseases for the bacteria or other virus. We need to use this model, this in vitro model, to analyze whether this compound candidate, this candidate compound can indeed generate a phenotype or toxicity to this, for example, the cancer cell, generate toxicity in the cancer cells. Okay, when we know this compound can inhibit the cancer cell survival, then of course we need to move to the animal study. We need to go into the preclinical study to test the efficacy and the safety in the animal, right? When we have this data, then we probably can move to the clinical trial, and in the end, we wait for the we can get a compound that can be approved by FDA, right? From the target selection to the clinical trial to the FDA approval, if you are lucky, probably we need to spend 15 to 20 years in an average. It's a really, really long time. So when you want to develop a drug, the first step and the second step are very, very important. It's very important to select what the target is important, and then the lead compound and also the data optimization become very crucial, because if these steps are wrong, then you will fail. You will spend a lot of time and the money when you enter the animal study or even the clinical trial. You will spend all the time, or you will all the experiment will fail. All the study will be failed. Okay, so during the drug development, there are two strategies to think about how we can get the candidate compound. Okay, the first strategy is we call the target discovery. It means that we already know which target we would like to focus on, and then, as I mentioned, that we develop an assay to test which compound can modulate this target activity. Okay, this is we call the target discovery, or we call the target-based screen. Okay, the second strategy is we call target deconvolution. This alternative, we can call it as the phenotypic-based screen. It means that we already have a drug, we already have the compound, we already have a candidate that has been shown to their efficacy in the specific phenotype. And then we have this compound, then we look for its target data. Okay, first we screen for a drug that can trigger the phenotype, and then we discover their target data. Okay, this two strategy has been used for many studies for the drug discovery. Yeah, and today's talk, I will include one target discovery strategy. Our drug discovery using the first one, target-based screen, and I will also show you how we move to the target deconvolution strategy. Yeah. So for the target discovery, the advantage of this strategy is the drug candidate's molecular mechanism has been understood, right? So because we need to know this candidate, their target is important for the disease development. Okay, and in this strategy, it's very typical. It's easy to be carried out, and it's less expensive, and the process is much faster. Yeah. And also because in this strategy, we already know the target, and we also develop the assay to analyze the target activity, right? So it's also easy for rationale design the new molecule with improved potency and safety. This is the advantage of the target discovery. Yeah, for example, another advantage is I would like to use this as an example. This is the signaling pathway from the receptor tyrosine kinase. One example is the EGFR. I think most people should know this signaling pathway already. So, and this signaling pathway has been well studied. Right? When receptor tyrosine kinase is activated, you will transduce the signaling to several downstream substrate, right? And this signaling will then induce different phenotype to modulate the cell behavior. For example, here, this part, the PI3K to AKT to mTOR, this pathway is very important to regulate the cell survival and cell cycle population. And for the cancer cell, this part is quite crucial. This part is quite crucial, right? This can help the cancer cell growth, right? And they also this pathway can also can also activate the RAF/MEK kinase to modulate the transcription and also the cell cycle population, right? And all this molecular mechanism has been understood. So because we already know this molecule, so we can choose some of them for our target. And in this strategy, we can also combine different inhibitors that target to different proteins in a different pathway. So, and this combination can generate the synergic toxicity to the cancer cells as well. So this is also the advantage for the target-based screening, drug screening. Okay. Okay, then I would like to go back to talk about the target disconvolution, the phenotypic based screen. And the advantage of this strategy is when we identify this compound, identify this candidate compound, we already know this compound has been shown the efficacy to the, we already know this compound indeed can generate the phenotype, can induce the phenotype. For example, we already know this compound can kill the cancers, right? Just we don't know which target it is, right? Right. So, but we already know this compound is really potent to kill the cancer, right? But the problem of this strategy is it will cost a lot of budget. And also you have to develop some special cell lines to discover such drug candidate. And the methodology is more complicated. And also it's much difficult to develop as a high throughput screening for this kind of drug. Yeah, this is the challenge. Okay. Yeah, as I mentioned, in this kind of strategy, we don't know the target. We can identify the target, this drug target later, right? But the important point I would like to show you that to know the target is essential for drug development. I think most people will think yes. Most people will think we need to know what target of this drug is. Otherwise, we cannot develop this drug. We need to know the mechanism of this drug, right? But actually, the answer is no. Yeah, because in principle, there's no regulatory requirement to know the molecular target of the drug or clinical candidate, because all that matters in the end is the drug need to be safe and the efficacy. Okay, so the drug needs to potent to kill the cancer, but it also needs to be safe for the normal tissues. Okay, this is really important for cancer drug development. Yeah, and actually, several approved cancer drug, the mechanism of the action is unknown when they were approved. Yeah, this table just listed for your reference. You can see that this drug has been approved for clinical use, right? But at that time, when they were approved, their molecular target is still unknown. Of course, right now we know that some of, for example, this vincristine is the target is a tubulin. Yeah, but at that moment when they are approved, we still don't know what the target it is. Okay, so just want to deliver one concept to you that for the anti-cancer drug, the most important thing is this drug need to kill the cancer and also need to be less toxic to the normal cells. This is the most important. Okay, and the molecular mechanism we can discover later, or yeah, actually it's not really essential. Yeah. Yeah, and for the high throughput, then I would like to talk about the high throughput. For the high throughput screening, we have to use this kind of the well plate, the multiple well plate, which we can test the compound. We can test a lot of compound in one plate. Yeah. So right now we already have the 9,600 well plate. So that means that in one plate we can screen 960 compounds. Yeah, we need to use this kind of a tool for the high throughput screening. Yeah, and high throughput screening means that one day we can screen 100 to 1,000 data. Okay, and another term is ultra high throughput screening, which we can screen over 10,000 data points per day. Yeah, this is just a definition. I will show you later. Okay. So to develop the high throughput screening, there are some key factors we need to concern. The first, all the screen in the whole procedure, the step need to be minimized. For example, it's better to have 2, 3, or 4 steps. If you have much more steps, then you will lose the screen result will much less precise and accurate. Okay, and we need to have a reliable readout for the plate reader. Yeah, and most important, the best high throughput screening is the mix and the read assay. It means that we just add a reagent and we can send the plate to the reader to get the result. If you can develop such kind of the assay, your result will more precise and accurate. You will more easy to reproduce your results. Okay, this is the key for develop the high throughput screening. Okay, after I introduce this concept, now I would like to move to our study. Okay, we know that to treat the cancer, the traditional chemotherapy is targeting to the factor that are essential for cancer proliferation. For example, gemcitabine. Gemcitabine is the drug that can block the nucleotide synthesis. Therefore, you can block the DNA synthesis to suppress the tumor growth, right? And the Taxol is a drug which can suppress the mitotic spindles formation. Therefore, Taxol can suppress the tumors, the cell splitting, right? Okay, so and this kind of the therapy, in general, they are really efficient to suppress the cancer, suppress the tumor growth. However, it also causes the high toxicity to the normal tissue. They are constantly growing and dividing, right? This is why later the target therapy has been developed. The target therapy is targeting to the growth promoting factors which are highly activated in cancer cells. For example, the Hesarpin, Hesarpin is the HER2 inhibitor, which HER2 is has been found overexpressed in many breast cancers. Is one biomarker of the breast cancers, right? Which can promote the cancer growth. And this kind of the drug also efficient, but they can only use to the specific subset of the cancer type. Okay, so in our lab, our dream is to develop a safer therapeutic strategy for a wide range of the cancers. Okay, so to this, we have cooperated with several chemistry labs, and we help them to analyze the mechanism of the action of the compound that has been designed and synthesized. Yeah, and we also focus on one protein target, TLK, and we cooperate with the chemistry lab to design the small molecule to inhibit the TLK activity. This part I will show you later. And also, we also develop some screening, high-throughput screening to discover the compound which has a potency to suppress the cancer growth or DNA replication. Yeah. So this is the study of in our lab. But today, yeah, I would like to focus on my long-term interest. The drug targeting to the chromatin replication, the DNA replication. Yeah. So, and sorry, I would like to move to another. Sorry, I need to move to another slide. Wait me a moment. Yeah. Yeah. As I mentioned before, yeah, I have spent a lot of time to study the mechanism of the DNA replication, right? Yeah, because I know that the DNA replication is the critical window for transmission of genetic and epigenetic information. Yeah, so it's not only the DNA will be replicated to daughter DNA, and also the whole chromatin structure will be disrupted during this process. And this chromatin structure will be reformed when DNA is replicated, right? So all the genetic and epigenetic information will be copied to the daughter DNA. Yeah. So, and because we have spent a lot of time to understand the mechanism of the DNA replication, we are particularly interesting to understand how the cell protect the genome integrity during this replication. We have particularly interested in this mechanism which can protect this genome integrity during this replication process. Yeah. So, and when I when I joined the TMU, I start thinking how we can use this knowledge to discover the cancer drug. So the first of all, we need to think about what the difference between the normal cells and the cancer cell in the replication process. Okay, so in the normal cell, the DNA replication need to be stimulated by mitogen. Okay, mitogen stimulation can trigger the scheduled DNA replication, and even this DNA replication is tightly controlled by a lot of the mechanism. Yeah, but even so, during this DNA replication, this normal scheduled DNA replication, there's still some error will be happen, and some stress will occur during the DNA replication. This we call the replication stress. And this stress will induce some DNA damage, very low level of the DNA damage. And this DNA damage can subsequently activate the checkpoint we call the sensor for the DNA damage. This checkpoint kinases when this checkpoint kinase is activated, they will recruit and activate the downstream DNA repair. And because this is a normal cell, and this DNA repair machinery are quite well, they can easily repair this DNA damage. They can easily repair this damage. So in the end, the cell can survive without any problem. This is the normal cell. In the contrary, in the cancer cell, we know in the cancer cell there are a lot of oncogene activation and tumor suppressor inactivation. This kind of appearance will cause unscheduled DNA replication. It will disrupt the tightly controlled, tightly regulated DNA replication. So the DNA replication will out of the control. And because of that, high replication stress will accumulate in the cells, and which will generate high level of the DNA damage. Okay, so this is why in the cancer cell, we can observe higher level of the DNA damage compared to the normal cell. And this DNA damage, again, they can also activate the checkpoint. And when checkpoint kinase is activated, they can also turn on the DNA repair to fix this DNA damage. However, the problem is in the cancer cell, most of the cancer cell has a deficiency in the DNA repair. So this cancer cell will in turn to activate the error-prone DNA repair. It means that this DNA repair system can fix the DNA damage, but you will remain a lot of error in the DNA structure or sequences. So in the end, the cancer cell can survive, but a lot of mutation and the genome instability will be accumulated in the cancer cell. As I mentioned here, they will have a lot of mutations. So therefore, they will in turn increase the oncogene activation and the tumor suppressor inactivation. This kind of the positive feedback loop will cause the cancer cell become more malignant. Okay, this is the current hypothesis why the cancer cell will become more, they will have a lot of mutation accumulation, and the cancer cell will be difficult to be treated, especially on the target therapy, right? Okay, and in this pathway, we can know that to survive, cancer cells are highly reliant on the checkpoint activity. Okay, they need the checkpoint kinase activity to activate the DNA repair system. Otherwise, they cannot survive. Yeah, this is why a lot of the there are several checkpoint kinase inhibitor has been developed here, and this inhibitor has been entered into the clinical trial. You can see here, right? However, so far there's no any checkpoint kinase inhibitor has been approved by FDA. And you can see this is the clinical trial results here. And you can see several clinical trial has been terminated. Yeah, and because of the toxicity. For example, in this clinical trial, this checkpoint inhibitor clinical trial, even this trial is complete, but they observe the severe cardiotoxicity as well. Okay, yeah, so this is why, this means that this kind of the checkpoint kinase inhibitor are also toxic to the normal tissues, right? So our idea is we know ATR, CHEK1, V1 are crucial to protect the genome integrity during the DNA replication. And they have been used as a target for the cancer drug development, right? We are thinking whether we can find other players they can also protect the duplicating genome integrity. So we can use this as a target to discover the drug. Okay, this is our initial idea. So this is a kind of the target-based screen. Okay, so we need to identify the target first. So to this, we have to establish a system that can detect unstable replication fork. We can detect the unstable structure genome during the replication. Okay, and to establish this system, we have to know when replication fork become unstable, what will happen. This is a normal DNA replication. So helicase is the enzyme which can unwind, open the double-strand DNA of the parental DNA, right? And then DNA polymerase will come to replicate the incorporate the nucleotide to replicate the DNA, right? So helicase will mediate the DNA unwinding, and the polymerase will add the nucleotide for the DNA replication. This is a normal replication. However, when this replication meets the challenge, for example, if there's no nucleotides, then replication need to be stopped, need to be slowed down, right? So polymerase will stop, but helicase will continue unwind the DNA. So what will happen? They will have the single-strand DNA accumulate at this structure here. Yeah, and this structure this structure will be very easy to be attacked by cellular nucleus, so it will become unstable. Okay, so we use this space. We develop a cell line which expresses GLP fused proteins, and this protein can specifically bind to the single-strand DNA. And so under microscope, after the hydroxyurea is a compound which can inhibit nucleotide synthesis. After hydroxyurea treatment, we can observe the GLP signal on the DNA. Okay, we can observe the GLP signal under microscope. Okay, we use this cell line to screen, to do the siRNA screen. siRNA screen means that we have the siRNA pool, which target to a lot of genes. Here we focus on the Genome library. The siRNA are targeting to the 572 genes which express the kinases. Okay, and after the transfection of this siRNA, we then do the pre-traction, and then we determine how many GLP positive cells remain on the slide. Okay, through this strategy, we identify V1 and the CHEK1. We identify V1 and CHEK1. These two well-known fork protector was identified. Okay, so these two factors can confirm our screen is working, right? And the top hit is a protein called the TLK, toxo-like kinase 2, and we have 3 siRNA give the positive results. Yeah, so therefore, what is the TLK? TLK, the full name is the toxo-like kinase 2. TLK is a kinase which can phosphorylate a protein called ASF1. ASF1 is a protein which is a histone chaperone, which can bring the newly synthesized histone from the cytosol to the nucleus, and then deliver these histone proteins to the replication fork to help the nucleosome formation assembly. Okay, So, and at that moment, we just know that AS1 can phosphorylate TLK can phosphorylate AS1, and AS1 is required for chromatin assembly during the DNA replication. But we still don't know whether TLK indeed has function to regulate DNA replication and the chromatin replication and chromatin assembly. Okay, so therefore we have to test whether when we knock down the TLK, whether replication will be affected. Okay, therefore we do this EDU incorporation assay. EDU is the thymidine arrow. You can see the structure is quite similar. Okay, we try to feed the cells with the EDU, and when the cells are replicating, they will incorporate EDU onto the DNA. So, and we can detect the EDU amount in the cells, right? So here there are a lot of cells, but only these 5 cells has the EDU signal, right? This means that these 5 cells are in replicating during replication, right? Okay. But when we knock down the TLK, we can see the signal from the EDU are quite low, very, very low. This indicates that TLK can regulate DNA replication, right? Because it reduces the EDU incorporation efficiency. Yeah. And in this in this study, we have also go into the detail to analyze the dynamic of the DNA replication. And we know that TLK2 can also regulate initiation and elongation steps. But here I would like to explain this because of time. I would like to introduce this technology in more detail. Just tell you the conclusion, TLK2 can regulate the replication initiation and also elongation steps. And we also do the micrococcus digestion analysis, and to demonstrate that TLK2 can also regulate chromatin assembly during the DNA replication. Okay, yeah, just tell you the conclusion. Just want to tell you that the TLK indeed can regulate DNA replication and the chromatin assembly. Yeah, and we also analyze the cell proliferation by colony formation assay, and you can see from the picture here, we can see the control cells can form the colony very well. But when the cell knock down the TLK, with the TLK knockdown, we can see the colony is quite difficult to be formed, right? So this data indicate that TLK2 is essential for the cancer cell growth. Okay, so here we made a conclusion that TLK2 can prevent the replication-dependent single-strand DNA accumulation. And TLK2 activity is required for DNA replication and the cell growth, right? And in another experiment, we also detect the high level of the gamma-2X in the TLK2 deplete knockdown cells. Gamma-2X is a marker for the DNA damage. So it means that when cell loses the DNA, when cell loses the TLK2 expression, the cell will generate the DNA damage. So the conclusion is TLK2 is also required for maintain the genome integrity during the DNA replication.
I'm sorry, Mr.
Yes.
Your slide is freezing on chromatin replication.
Sorry?
Your slide is freezing on chromatin replication.
Really? Okay, so and how about now?
Not yet, still freezing.
Okay, I will I will share the slide again. Sorry. Can you see again?
Okay.
Yes, sorry.
Yes, okay.
Okay. So, yeah. So we also detect the gamma H2S in the TLK2 depleted cells. Okay, so we therefore we made a conclusion that TLK2 is important for maintaining genome integrity during the DNA duplication. Yeah. So as I mentioned, that in the cancer cells, they will have a high replication stress, and the cancer cell will highly rely on the checkpoint activation for survival, right? So, and our study shows that when we knock down the TLK2, it will further increase the replication stress. So we were thinking whether in the TLK2 knockdown cell, they will become more sensitive to the checkpoint inhibitor. Okay, so we're just wondering whether when we knock down the TLK2, whether we can increase the sensitivity of the checkpoint inhibitor to the cancer cells. Yeah, so we try to treat the cells with the CHEK1 inhibitor, UCN01, and we can see that in the TLK2 knockdown cells, we can detect a high double-strand DNA break signaling, like here. And we also visualize the DNA break by pulse field gel electrophoresis. We can see a lot of DNA break accumulate in the cells with the TLK2 knockdown and the CHEK1 inhibition. Yeah. Of course, we also test the cell viability, and we can see that in the cells treated with the viability of the cells with the TLK2 knockdown is reduced a lot when they treat with the UCN1, the CHEK1 inhibitor. Okay, so it means that TLK2 depletion can make the cells more sensitive to the checkpoint inactivation. Yeah, this is so our conclusion is that when we knock down the TLK2 cells, and the cell become highly sensitive to the checkpoint inactivation, and when these cells treated with the checkpoint inhibitor, the cell will this kind of the treatment will trigger genome catastrophe, which will lead the cancer cell die. Okay, and this data has been published on the Science Advance in the 2018. If you are interested, there are a lot of data I didn't show you today. If you are interested, you can have a look at this paper. But I would just this paper tell us that this study tell us that TLK is the potential target for the cancer treatment for our cancer drug development, right? So we cooperate with the Dr. Xuanli in the School of Pharmacy, TMU, and we would like to design the inhibitor because so far there's no TLK inhibitor has been developed. There are some compounds they show to inhibit the TLK, but their activity is still controversial. Okay, so we cooperate with the Dr. Xuanli to develop the TLK inhibitor. So our advantage is that we know the TLK can phosphorylate the AS1. AS1 is a substrate of the TLK. And we have the antibody that can again recognize this phosphorylation. So we use this to screen the compound synthesized by Dr. Xuanli, and we identify that through several compounds, we identify 6 compounds that can significantly reduce the AS1 phosphorylation. Okay, and we test the toxicity of this compound. As I say that the most important for the cancer drug development is the toxicity to the cancer, right? So of course we need to measure the toxicity to on the cancers. So we test their toxicity. We find that 6 of these compounds that can inhibit the TLK, they showed 5 of them show the higher toxicity to the cancers, significant toxicity to the cancers. Yeah. So therefore, we analyze, we need to make sure whether this compound can induce the phenotype as the TLK depletion knockdown, right? So of course, we need to know whether this TLK inhibitor can suppress the DNA duplication. So we do the EDU incorporation assay again, and you can see that this compound can suppress the DNA duplication in dose-dependent manner. Okay, there are two compounds, for example. And we also find that this compound can trigger the significant DNA damage in the cells, in the cancer cells here. Okay, yeah. And okay, sorry. This phenotype, so this data demonstrate that TLK inhibitor can phenocopy the TLK2 knockdown phenotype. Okay. So then we also test the different cancer cell line. Here, the MDA-MB231 is the breast cancer cell line, and A546-49 is the lung cancer, and HCT116 is the colon cancers. And we tested this cancer cell line, the toxicity on this cancer cell line. And the value I show you here is the IC50 value. And it means that the concentration, they can suppress 50% of the cancer growth. Okay, so we can see that this compound show the micromolar toxicity to the 3 different cancer cell lines. But in the normal lung fibroblasts, IMR 90 and MRC5, we found that these fibroblasts are less sensitive to these compounds. So in this data demonstrate that our compound is more, specifically toxic to the cancers. Yeah. And again, this study has been published last year. Yeah, and also I have several data I have not shown here. Yeah, if you are interested, you can have a look at this ECMC paper. Yeah, yeah, and until now, all this study we explored the target-based screening strategy to discover the anti-cancer drug. This is the strategy I mentioned, the first strategy I mentioned earlier, right? So, and we identified TLK2, which can protect the duplication genome integrity, right? And we use this as a target to develop the cancer drug. Yeah. And we are thinking whether we can use the second strategy, the target deconvolution screening, to discover the anti-cancer drugs. Okay, our idea is, so in our previous study, we used the TLK knockdown or TLK inhibitor. It can trigger higher duplication stress in the cancer cells. And then we can combine the checkpoint inhibitor to trigger the synergically toxicity to the cancer cells, right? I'm thinking whether we can change the siRNA to the compound library. We can use the compound, we can search for the compound which can increase duplication stress. And then we can combine the checkpoint inhibitor to trigger the synthetic lethality in the cancer cells. This is the idea. Okay, this is the idea. So we, instead of using the siRNA, we change to the compound library. Yeah. So this is our scheme that, first of all, we would like to use the high throughput chemical screening to search for agents that can trigger duplication stress. Okay, this is our step to see which compound can increase the replication stress. And then we would like to combine this compound with the checkpoint kinase inhibitor to see whether this compound can generate a synthetic lethality to the cancer cells. And then because of the, then we can move to the mouse model to test the anti-tumor efficacy and the safety. And of course, as I mentioned, we can determine the mechanism of the action later, right? Okay, this is the our plan. Yeah, and actually we have done this already, the first one of this study already. Yeah. So to perform this, to execute this project, we use this reporter cell line, this GLP reporter cell line, to determine which compound can trigger replication stress. Okay, if the compound can trigger replication stress, we can detect the GLP signal on the chromatin. Yeah. So in this study, we cooperate with the Professor Huang Wei Zhan. He is also the chemistry expert. We asked him for the compound library, and he just gave me he provide 160 compounds to us, and we try to screen this compound with our GLP reporter cell line. And you can see here the blue dots means that the compound, they can increase the GLP signal in this cell line. Okay, so therefore this compound are our compound candidates, right? So to make sure this is to validate this compound is our candidate, we need to know whether this compound can suppress DNA replication. Again, we go back to the EDU incorporation assay, and we pick up 3 compounds as an example here, and we can see these 3 compounds significantly suppress the DNA replication. So they are replication inhibitor as well, right? Yeah. So we go to the next step. We try to combine with the checkpoint kinase inhibitor to see whether they can generate synthetic lethal. Yeah. And we use the U2OS, this osteosarcoma cells, for first run test. And this is, first we test one of the checkpoint inhibitor, AZD7762, which is the CHEK1/2 inhibitor, CHEK1 and the CHEK2 inhibitor. Okay, so you can, and the Y-X means the cell survival. So you can see here that when we treat the AZD, this CHEK1 inhibitor, CHEK1/2 inhibitor at 50 nanomolar, the cell viability seems not reduced a lot, right? Not significantly reduce their viability, right? But when we combine with our compound 161, you can see that the cell survival reduced to only 10%, only 10%. Yeah, and this reduction is a dose dependent as well. Yeah, and this is a CHEK1, CHEK2 inhibitor. We have also tested the CHEK1 specific inhibitor, and WEE1 specific inhibitor, and also ATR inhibitor. And we got a similar result from all these inhibitors. Yeah. In addition to the 161, we also test the compound 19, and we got a similar result as well. And also, we also test other cell line. This is MDA-MB231 cells. This is triple negative breast cancers. You can see that the data are quite similar, right? And another cancer cell line, colon cancer DLD1 cells, and non-small cell lung cancer H1299, we can also get the similar results. So this indicates that this combination treatment can trigger the synergic toxicity to a broad range of the cancer cell lines, right? Okay. So as I mentioned, next step is we would like, as I mentioned, that combination with the checkpoint kinase inhibitor may generate a genome catastrophe, which could lead cancer cell die, right? So we want to make sure that whether this combination indeed can trigger the genome catastrophe. So we determine the DNA damage, double-strand DNA break here. We can see that the red box means that the combination treatment. Okay, we can see that combination with the CHEK1, CHEK2 inhibitor or the V1 inhibitor, we can see the significant double-strand DNA break signaling activated in these cells. And we also confirm the double-strand DNA breaks by pulse-field gel electrophoresis. We can observe the heavy DNA breaks in this combination combined treated cells. Okay. And regarding, so next we would like to test the toxicity to the normal cells. Here we use the normal epithelial cells. So we can see this blue line means the toxicity, the cell viability of the normal cells. And we can see this, the combination of our compound with the ADD7762 did not generate high toxicity in the normal epithelial cells here. Okay. So when we look at the cell proliferation, so the y-axis here is the viable cell number. So if you look at this, the number of viable cells, yeah, and sorry, in this experiment, we only treated compounds for 24 hours. And then we remove the compounds and let the cells continue grow for several days. Okay, so we can see that the combination treatment, these two lines, green and the red, means the combination of treatment. We can see that combination of treatment almost block the cell proliferation, right? But in the normal cells, combination of treatment will suppress the cell proliferation during the treatment, this 24 hours. But later, when we remove the drugs, we can see that cell proliferation resume and continue grow without stop, right? So, and when we look at the dead cell percentage, the death rate, the dead cells percentage, we can see that combination of treatment in the cancer cell will increase a lot high percentage of the dead cells, but in the normal epithelial cells, it did not increase much the cell deaths. Okay, so this data tell us that combination of treatment are specific toxic to the cancer cell lines. And we look at the DNA damage. The combination of treatment trigger high level of the DNA damage in the cancer cells, but in the normal cell, only weak DNA damage can be observed. When we look at the apoptosis event, we can see that combination of treatment can trigger the DNA, cell apoptosis in the cancer cells, but not in the normal cells. Okay, confirm the toxicity is specifically to the cancer cells. Okay, then we move to the mouse model, and this mouse experiment will cooperate with the Dr. Stan Gerson in the Case Western University. Yeah, and they perform the animal study, the animals model to us, and in their study they found that this is a two cell line based nail graph. So you can see that combinational treatment significantly suppress the tumor tumor growth in both radiograph models, right? But this treatment did not significantly affect the body weight, mouse body weight. So suggests that has a low toxicity, no side effect to the mouse. Okay, and we also isolate the patient pancreas tumor. We also isolate the pancreas tumor from the patients and inoculate into the mice and measure the tumor's growth. Yeah, so as you can see that our combinational treatment also significantly suppress this tumor growth in this PDX model. Okay. We also isolate the tumor from these mice, and we can see that this is a quantification data for the DNA damage. We can see that combinational treatment induce the high levels of DNA damage in these tumors. Okay, but when we look at the mouse tissues, kidney, liver, and the lung, we did not detect significant DNA damage from these normal tissues. Okay, so this data tells us that the combinational treatment might produce the very weak side effect to the mouse animal. Yeah. Okay. So because of this success, right? So in our study, we only tested 160 compounds for the initial screening, right? And it looks like all the screening platforms are working. And yeah, so we try to discuss with the Dr. Winda in Genomics Research Center of Academia Sinica. We try to discuss with him to whether we can do the high throughput screening. No, ultra high throughput screening. In the Academia Sinica, they have over 2 million compounds library. So we are trying to see, we discuss with Dr. Wu to see where we can do the screening on our reporter cell line. Yeah. So in our screen, we use the 384 well plates, and each plate we can screen around 300, and each round we can screen 30 plates. So it means that each round we can screen over 10,000 compounds. So this is why we call this is ultra high throughput screening. Yeah. And because of time, I cannot show you the video how we do this, but just tell you that we have all the robotic machine that can help us to add the compound from this white plate to our cells. Okay, so we need this kind of, because the compound, we only can add 0.2 microliter of the compound to the cells. So it's impossible to do it manually. Okay, and we also have the automatic robot to do all the process of the screening, but we only have a 4 step. If during this high throughput screening, we only have 4 steps, so we can get the result within 1 day. Yeah, and this is the end of the data, the example for the data. So you can see that this is the GLP signal in the control cells, and this is the hydroxyurea treated cells. We can detect high level of the GLP signal from the partially controlled hydroxyurea treated cells, right? And this is a cell nucleus, okay? And with this kind of the screening, we aim to screen 2 million compounds. But right now, we only screen 150,000 compounds. This is already done. This has been done in the academia clinical. So this is some example from the 7,000 compounds. You can see that we already identified a lot of compound candidates for future study. Yeah. So I would like to close my talk here, and I would like to I would like to mention one thing, that most of work I present today for the drug discovery were done by my PhD student, Tingyu Zhang. Okay. And I also want to thank Dr. Xueyun Li and Wei Zhan Huang, and also in the TMU, and also the Dr. Gerson in the Cass Western, and Dr. Yan, his postdoc, who did the animal study, most animal studies. And also thanks my previous advisor, Anya in BRIC, and Lilong in GRC. And also thanks Dr. Wu for the high-throughput screening. So, and thank you for your listening. I'm happy to take the question.
Okay, thank you very much for Mr. Sung Bauli for the very insightful presentation. Now we are going to continue with the question and answer session. I saw that there is one question, from Bu Erna, in the chat room. I will read the question. How to make sure the compounds may be from natural substances, as the LK inhibitor only work to cancer cells, not to normal cells. What is the parameter to make sure the safety of the active compounds? So how that it will work on the natural substances and not to the cancer cell, not for the normal cells.
Yeah, I think this is quite good question. I mean, for the cancer drug development, this is really important. I mean, the efficacy and the safety, right? So in the cell-based level, we have to test. We actually in my study, we also test the lung fibroblast, normal fibroblast. We have to know this compound only generate very low toxicity to the normal cells. This is first step. And yeah, but sometimes I have to say, if your compound is toxic to the cancer cells, especially to a broad range of the cancer cells, it probably can also show some toxicity to the normal cells. But the window need to be high, need to be large. We need to increase the window between the normal cells and the cancer cells. For example, in our study, our TLK inhibitor shows the IC50 around 0.5 nanomolar, micromolar. But actually in the normal fibroblast, it will be it's better to higher than 5 or 10 micromolar. If you have this kind of a window, it will be more safe. Yeah, but no matter what, to determine the safety, we always need to go into the animal model. We always need to analyze whether our compound is toxic to the animal or rat, even the monkey. Some people will use the monkey because you're more related to the human. Yeah, we have to make sure that they didn't generate a side effect in this kind of the model. Yeah, and today I will only show the some IHCs. Sorry, let me go back to in this. Today I only show this IHC staining, but actually we also monitor. We also monitor the some common side effect they can observe in the chemotherapy patients. For example, diarrhea, because of the GI system is affected. And we also monitor the hair loss, right? And yeah, we did not see any we did not see any effect on this kind of the during this drug treatment. Yeah, so it's not only monitor this kind of the DNA damage. We also need to see whether this mice is grows well, healthy. Yeah, this is how we have to determine the safety.
Okay.
I don't know whether I answered the question.
Okay, so the answer will be the test experiment first on the animal, on the monkey especially.
Yeah.
Okay.
Yeah. Yeah, because when our drug want to go into the clinical trial test, they always need to see the animal data. So we cannot jump this preclinical study. So the safety is measured in the beginning is measured in the animal, in the mice, and or other rat or other animals. Yeah.
Okay, thank you, Mr. Sungawi, for the answer. I have also a question from Mr. Sentot, yeah, from the personal chat. We are interested in the screening of the anti-cancer compounds. From some of the compounds that can be used to fix the mutation is the compounds number 161 and number 19. How is the treatment of this compound? Is it isolated or in the form of extract? And then how is the protocol of the treatment, yeah, to prove that the compounds has the anti-cancer ability?
Sorry, I did not really clear your question. You are asking how to know this toxicity, or
how is the treatment of the compound so that the compound can be used as the anti-cancer to prove it as the anti-cancer has the anti-cancer ability?
Okay. This is also the question I have to say, because it pointed our some strategy. So I try to hide it, but I will tell you now. Yeah. So in our cell-based experiment, for the TLK in the previous study, probably I can show you. In this TLK inhibitor, we treat these cancer cells for 3 days, for 72 hours. Okay, we just directly treat the cells. We just put our compound in the medium and analyze their how many cells survive, how the percentage of the survival cells. Okay, by this MTT assay. Yeah. And in this study, we treat the compound for 3 days, and the data is looks like this. However, in this study here, for the combinational treatment, I have to say we first try to treat this compound for 3 days, but unfortunately, at that moment, we did not see the synergy effect. We did not see any synergy effect. Yeah, I think that's because the single treatment gives the higher toxicity at this condition. Yeah. So why we did this, I can show you the scheme here. It's like a proliferation assay, like here. Yeah. So we try to treat the cells only for 24 hours, and then we remove the drugs, and then the cells continue to grow for 3 days. And this is, yeah, as you can see from this growth curve, this kind of the treatment the combination treatment can significantly block the cell proliferation. But for the single treatment or for the untreated group, there's no any effect on the cell growth, right? So we use this to determine the toxicity. So I cannot tell you which strategy to determine the toxicity is the best. We always need to test the condition. Okay.
Okay, thank you. Is there any other questions from the audiences? Maybe if you want to ask directly, you can also use the feature of raising hand. Or perhaps you can also write your question in the chat room and I will read it for you. Okay, is there any more question? Okay, there is also a question from Bu Karin, Miss Karin. Okay. When is the cancer cell get mutated? Even though we have already treatment, yeah, and already mentioned that we have already said that we are cured from it, but in some few more months, it is found the new cancer cell. So how can we know that the cell got some mutation in the body, especially? That is the question.
So which question? I did not see. Is in the trial or?
No, the question is through my WhatsApp. Yeah. So
okay, sorry.
Yeah.
Sorry. So can you
yeah.
pardon again?
So the question will be, when is the cancer cell get mutated inside the body? Because some facts shows that someone is already treated and being cured from cancer, yeah? But then again, after a few months, yeah, the person is honest that there is still cancer cell left in the body. Yeah.
Yeah. Okay. So I don't know where my answer is correct, but I would like to say my thought is when the patient treated with the anti-cancer drugs, no matter which one is targeting or chemotherapy, this kind of drug, you can imagine that only the cells located on outside of the tumor can expose this kind of drug, right? So this the cancer drug treatment can only kill the outside cells of the tumors. So, but in our view, we can only see the tumor become smaller and smaller, unless we cannot detect it. But when we say the tumor is cured, actually, it just means that the tumor is become small to be invisible. It's not real kill completely. Okay, so I think this is why after a few months or few years, you will see that the tumor become reverse again, grow again, and even become more malignant, spread to other tissues. It will be more difficult to be treated. As I mentioned in the earlier that's because the mutation is accumulating more and more and more. And the tumor is difficult to treat, especially the target therapy. Yeah.
Okay, thank you. So the mutation will always happen, and the cancer is not fully cured. Yeah, only getting smaller. Yeah. Okay, thank you. There is also a question from Malinda, yeah, in the chat room. I want to ask about the risk of drugs in the table on slide 13. of the first material. Yeah.
Oh, first material. Okay.
First material. I think about the pharmaceutical. Okay. There are several drugs used for the same type of cancer. What is the difference between the types of drugs and their treatment for the cancer? Are all types of drugs with the same function and different types still being used today? Thank you. Yeah. So what are the difference of the drugs mentioned there?
Oh, you mean the drug I mentioned in these tables?
Yeah, I think so.
Actually, the mechanism of action of this drug are totally different. For example, you can see, I think some of you already know that doxorubicin. Doxorubicin is the drug they target to the topoisomerase, right? And for example, gemcitabine, as I mentioned in my talk, that targeting to the nucleotide synthesis. And also cisplatin is how to say, interlinking, you will incorporate into the DNA to generate the DNA damage. So all the mechanism of this compound are quite totally different. The table showing here is just want to tell you when this drug has been approved, which year this drug has been approved, and for which cancer type. Okay, so they are not really correlated. Yeah.
Yeah, so the table only shown about the year, the year the drug is being discovered.
Yeah.
And all
I show you this table is just want to tell you that at that moment when that has been approved by FDA, at that moment their molecular target here, their molecular target is totally unknown. NT means the non-determined. Okay, so no target, and they just know that this compound can sufficient to kill the tumor. Okay, to reduce the tumor formation in the animal and also pass through the 3 clinical trial. Yeah, but at that moment when they approved, they didn't know which target they belongs to. Yeah.
Okay, thank you, Mr. And also there is one more question, from Bukarin, I think already been answered. From Mas Hafid, from Mr. Hafid. The question is about the anti-cancer drug. Is it only for those who have like cancer problem, or is it also for severe cancer treatment for the drug itself? And how feasible the society can get this kind of drug? How easy the society can obtain the drug?
I don't really understand the key point for this question.
About anti-cancer drugs in the society, whether it is for cancer that already in the first stadium, or it is also for severe cancer, usually drug itself work.
Actually, I think most of people may know that if we can treat the patients at the very beginning when the tumor is happen, it will be more easy to cure the tumors. Yeah, but however, in most of the case, the patient will know they have a tumor grow in the body. It's already over phase 3, even phase 4. The tumor is become very big or spread to other meta to other tissues. So this is why in most of the clinical trial, if they want to test the efficacy first, they will choose the phase 3 patients because it will be more easy to measure the suppression of the tumor growth. Yeah, and there will be more cases for this one. Yeah, but if we want to treat a case, I would say it would be better to treat the patients as early as possible. The possibility to completely cure the cancer would be higher, much, much higher. Yeah, but it's really, really not easy. Yeah.
Okay, thank you. Okay, I think that's the question. Yeah, I think that's all the questions that being asked by the audiences, and I think that the time is already up. Yeah, so I think I need to close this session. Thank you very much for Mr. Sumbauli, yeah, for the information, for the very insightful presentation. And also thanks for the audiences for being active in asking questions. Hopefully all the question has already been answered nicely. Okay, I think that's all. I give it back to the MC, Ms. Andini. Thank you.
Thank you very much.
Okay, thank you so much to Mr. Tian that has helped us to moderate this presentation of the seminar, and big thanks to Lao She, Li Shengpao for the information and the knowledge which have been given to us. May it useful for us, and it totally will be so useful for us. Okay. We finally come to the end of today's agenda. So thank you for everyone participation for today's lessons. Yeah. So Prof. Li Shengpao said that target, there are still many opportunities because of the 2 million target components, only 150,000 components that are known. That's right. So there are still a lot of opportunities for us to search or to seek the agent for more like that. Yes, Mr. Prof. Okay. Okay, before we close our seminar today, I would like to inform that if there's any closing remark from Prof. Lee, please welcome.
Yeah, Mr. Lee, maybe you have some closing remark, yeah, to end this meeting, maybe something to say.
Okay. Yeah, thank you very much. I'm a bit nervous for these lectures. Yeah, this is I don't know. There's a lot of the audience who will come to join this one. Yeah, and thank you all of you for the invitation and also attentions. I would say we have a lot of the idea and also a lot of the drug candidate need to be searched. And of course, we are welcome to cooperate with all of you. And if your students like to join the TMU to work on the similar topic, no matter it's the medicinal chemistry or it's about our like cell biology fields, yeah, you are welcome to contact me. Yeah, you can just send me a mail. Yeah, I can try my best to help you. And thank you very much. Yeah.
Ya, thank you Mr. Sumbauli ya. Jadi teman-teman kalau misalnya ada yang tertarik melakukan penelitian tentang cancer ya, ataupun juga ada yang tertarik ingin mengikuti seminar, webinar tentang cancer, you can contact, bisa menghubungi langsung melalui email Mr. Sumbauli ya. Nanti emailnya akan diberikan oleh panitia. And also maybe we can perhaps we also can get the material from today's seminar ya, Mr. Sumbauli ya. Maybe later we will contact for the material. Okay, thank you.
Okay, once again, thank you for every participants for today's lessons. Let's us pray together before we finish the seminar. Thank you, God Almighty, for allowing us to meet using Zoom to have another lesson. Although we cannot face to face in class, we pray that everything that we have learned today will bring great impact for all of us. Please, God, bless us, bless all of us, our lecturer, our moderator, and everyone that has helped us today. In God's name we pray, Amen. Those are the agendas we have presented to you all this day. We hope you learned something. As a master of ceremony, thanks to launcher Li Shengbao as the speaker, Mr. Kian as moderator, Mrs. Ambar, the committee, and the audience entirely. May what we had conducted will be useful for us. Thank you so much for your attention. See you on the next occasion. Have a good day, everyone. 谢谢老师。
Thank you all.
Thank you very much.
Thank you very much.
Thank you very much, Prof.
Yeah.
Sudah foto bersama ya Bu Andini.
Oh yeah. We would like to take a online picture. Is that okay, 老师?
Buoro, sayu buoro. Adakah buoro?
Oke, kita akan take a pic. Jadi nanti silakan admin untuk mendokumentasikan kegiatan kita pada siang hari ini. Bisa dikasih apa-apa ya Bu Ayu? Like 1, 2, 3 cheese gitu. Bu Ayu, monitor? Bu Ayu atau Bu Ambar?
Kameranya mohon dibuka,
Oh, iya.
teman-teman mahasiswa. Kameranya di-on-kan dulu. Untuk teman-teman mahasiswa, kameranya di-on-kan dulu. Kita foto bersama. Diberi kode mungkin Bu Andini.
Ya, oke. 1, 2, 3, 1, 2, 3 cheese. Oke, sebentar. Next page. 1, 2, 3 cheese. Okay. Next page. We have a lot of page. Okay. Keep smile ya. 2 page to go. The last one. Okay, we've done. Thank you so much once again, Prof. Lee. Thank you so much for all the participants. Thank you, Ms. Ambar. Thank you, Ms. Kiantoro. See you in the next great opportunity.
Okay, thank you, Miss Andini. Thank you very much, Prof. Lee. We apologize if there are some apologies things, Prof. Thank you so much.